Okay, everyone, so now we're going to move on to Chapter 2, mitosis and meiosis. This should be a review for most of you. You've had general biology, so you should have covered cell division in biology. We may hit on some new topics, but for the most part, you guys should already know most of this stuff. So hopefully we'll get through it pretty quickly. So just a little bit about review about DNA and chromosomes. Like we said before, we now know that the genetic information is found on chromosomes and is in the form of DNA. And of course we know that that DNA has to be passed on to daughter cells through the process of cell division. And the two types of ways that we do that is through mitosis, which is the division of somatic cells, or everyday cells, your skin cells and your liver cells and your blood cells, red blood cells, all those divide through mitosis. When we produce sex cells, it's important to reduce the number of chromosomes by half so that when we form a zygote through the process of sexual reproduction, we don't have too much DNA, and that process is called meiosis. So just a little bit more detail about meiosis and mitosis. We know that this is cell division. Mitosis will lead to the production of two identical cells, and those two identical cells will also be identical to the original parent cell and will all have the same number of chromosomes. Meiosis is going to be a little bit different. The genetic material is reduced by half, and this is important, like I said, because we don't want to double the genetic material every time we produce progeny. So just talk a little bit about cell structure. We know there are two types of cells. You have eukaryotic cells and prokaryotic cells, and this is just a picture that I found on the internet. And shows you the difference in the two structures. The main critical points are the eukaryotic cell has a nucleus and the prokaryotic cell does not, and the eukaryotic cell is more compartmentalized, and so it has membranous organelles like the Golgi apparatus, endoplasmic reticulum, mitochondria. You probably went into a lot of detail about all these cell structures in general biology, and we're not going to do that in this class. We're just going to focus on the cell structures that are important for genetics. Of course, the prokaryotic cell doesn't have all those structures, but it does have the plasma membrane. It does have, of course, its DNA genome. It does have ribosomes for synthesizing protein. It's just not as sophisticated structurally as a eukaryotic cell. So just a little bit more detail about the plasma membrane. We know it's made up of a phospholipid bilayer, and of course it has proteins associated with it as well, and it's going to control how materials move in and out of the cell. Plant cells have an additional outer cell wall made of cellulose, and many animal cells have an outer covering or cell coat called the glycocalyx that's going to have a lot of proteins and carbohydrates and other molecules that are part of cell signaling. So just a little more detail about eukaryotic cells. We know they contain a nucleus, which is the membrane structure that houses the genetic material and keeps it compartmentalized from the rest of the cell. Also within the nucleus is a structure called the nucleolus. This isn't really a structure, it's more just a region of the nucleus that stains heavily because it's got a lot of proteins, because it's the site of ribosomal RNA synthesis. And ribosomes have a lot of proteins, so as the ribosomes are being built and assembled, that part of the cell is going to stain darker, and so it appears as a structure. So this is the picture of the eukaryotic cell in your book, and you can see here's the nucleus and this little heavily stained structure here in the center is the nucleolus. Now we have mitochondria. These are these little structures here. They're the site of ATP synthesis, so this is where glucose is brought in. Well, the glucose is brought into the cell, into the cytoplasm, and is broken down into 2-pyruvate, which then enters the mitochondria to form ATP molecules. The endoplasmic reticulum, you can see here, there's two forms of endoplasmic reticulum. You have what's called the smooth endoplasmic reticulum, that's where lipids are made, and then the rough endoplasmic reticulum contains ribosomes. So all these little dots here on the endoplasmic reticulum are the ribosomes, and so that's going to be a site of protein synthesis. Now, ribosomes can be found scattered throughout the cytoplasm, and you know, proteins are also made there, but proteins that have to be processed significantly, they have to have things added to them. Like, for example, if you've got a protein that's on the surface of the cell that has a carbohydrate attached to it for cell signaling, that protein is probably going to be made on the rough endoplasmic reticulum, and then those changes, that carbohydrate is going to be added to it in the endoplasmic reticulum. The Golgi apparatus is here, this structure here, and the Golgi apparatus is basically the UPS of the cell for proteins. So as proteins are made either in the cytoplasm or the endoplasmic reticulum, they will travel to the Golgi body and kind of get packaged and processed, and from there they'll get shipped to wherever it is they're supposed to be. So if it's a cell membrane protein, it'll go to the plasma membrane. If it's cytoplasmic enzyme, it'll stay in the cytoplasm. If it's something that needs to go to the nucleus, like it's maybe a enzyme involved in replication of the DNA, then it'll get transported to the nucleus. And so that's how the Golgi apparatus works, or sometimes it's also called the Golgi body. And chloroplasts are found only in plants, and this is the site, or not only plants, but photosynthetic organisms, so plants, algae, and even some protozoans. And this is the site of photosynthesis. So this is where it takes light, energy, water, and carbon dioxide and produces glucose molecules. The centrioles are pictured here in the cell. And these are very important for the construction of the spindle apparatus, which we will talk about shortly. It's located in a region of the cell called the centrosome, and so it's very important for cell division. And then, and also the centrioles are composed of microtubules, which are made of the protein tubulin. Okay, so in contrast, prokaryotic cells are a lot simpler. They don't have a nucleus. They don't have any of those membranous organelles. Their DNA is attached to the cell membrane, and so it kind of sticks to a particular region of the cell, and that region is called the nucleoid region, and it's pictured here in the picture. But the nucleoid region is just kind of a designated region. It's not like the nucleus where it's a structure. The prokaryotic cells don't have a nucleolus, but they do manufacture ribosomal RNA and produce ribosomes. So they do have, like I said before, they do have a membrane, they do have ribosomes. Generally they have a cell wall. For bacteria, it's usually made of peptidoglycan, and then they'll have some extra structures depending on the type of bacteria you're talking about. So some have flagella, or these other little hair structures are called pili. This picture here is typically what a E. coli would look like. So we've talked a little bit about DNA and how it's found in the form of chromatin, which is basically DNA complex with proteins. And when we get to the chapter where we talk about how DNA is packaged into chromosomes later in the semester, we'll talk more about those proteins and how specifically they complex with the DNA. But when you look at it in a microscope, the chromatin just looks kind of like a diffuse mess of stringy strands. You can't really see any individual chromosomes. But when the cells are getting to the point where they're getting ready to divide, that's when the cells condense into the chromosome structures. And so you can actually visualize individual chromosomes. So the spindle apparatus is going to be really important for how chromosomes move during cell division. The centrioles, or those structures we talked about before, they're located in the centrosome, and they're going to be the site that organizes the spindle fibers. And so you can see here, this is part of the spindle apparatus. This orange structure here in the middle is the centrioles, and you can see all the spindle fibers kind of extending out from that region. In the middle here, the blue color, that's the actual chromosomes. And I believe those individual dots are supposed to be representing possibly the centromeres of the chromosomes. But you can see there's two poles to the cell, so this is the other side, this is the other pair of centrioles, and you can see the spindle apparatus kind of extending out from there, and you can see how the chromosomes in the middle here attach to these spindle fibers. So the spindle fibers are really important for organizing the chromosomes and moving them during the process of cell division. And so you can see here in this picture how the actual spindle fibers attach to the chromosome. And you can see they attach to this region here in the center, and that's the centromere. Now each chromosome has a centromere, and the location of the centromere can vary chromosome to chromosome. So the centromere is this region of the chromosome that attaches to the spindle fibers. And extending from the centromere, you'll have what we call arms of the chromosome. And a lot of times the centromere is not dead center. So if it's not dead center, then the shorter arm is designated as the P arm, or the petite arm, and the longer arm is designated as the Q arm. But chromosomes can vary for centromere position. If they are dead center, they're called metacentric. If they are kind of towards the center, but you do have a shorter arm and a longer arm, they're considered submetacentric. If you have a situation where the short arm, the P arm, is extremely tiny, that's called acrocentric. And in some cases, the centromere is actually at one end of the chromosome, and in that case, it's called telocentric. So as we've discussed before, chromosomes are found in pairs. Those pairs are called homologous pairs. So the two chromosomes are the homologous chromosomes. And so you can see here, this is depicting, you have two individual homologous chromosomes. All chromosomes during the life cycle of a cell will replicate themselves before the cell divides. And after that, you still have individual chromosomes, but now they have duplicate the number of DNA, the amount of DNA with those chromosomes. And so in this case, you have two sister chromatids that make up one chromosome. And so you have 4 times. So for example, if you have a gene that codes for hair color, and let's say it's, the particular allele is brown hair. So we'll say that it's the same for both chromosomes. So right here you have two copies of the gene for, here you have 2 copies of the gene for brown hair. After replication, you actually have, now you have 4 copies of the gene for brown hair, but you still only have 2 chromosomes, because even though it's got double the amount of DNA, it's held together by a single centromere, and this structure here is still considered one chromosome made up of 2 sister chromatids. So where we get these chromosomes from, with each pair of homologs, one came from the mother, so we'll say this one is from the mother, and one came from the father. Let's say the green one came from the father. And like I said, both of these homologs are going to double, they're going to replicate themselves before they divide. So you end up with 2 copies of each chromosome held together by a single centromere, and those copies are referred to as sister chromatids. So as far as chromosome number goes, again, this is just review. We've talked about this before, but somatic cells or diploid cells are always going to have a specific number of chromosomes. For example, humans have 46 chromosomes. Those chromosomes are present as homologous pairs because you get one set from your mom and one set from your dad. So we have 23 homologous pairs. Mice have 40 chromosomes with 20 homologous pairs, just as another example. So homologous chromosomes carry similar genes. So they'll carry the gene, one chromosome might carry the gene for hair color, and so its homolog will also have a gene for hair color, like we said before. However, they're not identical because there are different versions of the same genes that are present in the population, and those versions are called alleles. And we'll talk a lot more about that in Chapter 3. So this is a karyotype, and a karyotype is just a picture of all the chromosomes in a cell when they're condensed down to the most condensed point where they're visible during cell division. And so what they can do with a karyotype is they can stain the chromosomes and take a picture, and then they can use cut and paste to line them all up side by side. And so you can figure out which chromosomes are homologous because they're the same size and they stain the same way, and so you can pair them up. And so here you can see the human karyotype with 23 pairs. However, note here, this pair does not look homologous. This is the XY, so this is a male karyotype. And we know males have one X chromosome and one Y chromosome. These aren't strictly homologs because they carry different genes. However, they do pair up during cell division, and there are some similar areas of the 2 chromosomes that allow them to pair up. But the true homologous pair would be in a female where we have 2 X chromosomes. Alright, so just a little bit more about alleles. Like I said, the homologous chromosomes are nearly identical in size and their location of genes, but the actual genes that they carry can vary. Of course, the diploid organism is going to contain 2 copies of each gene. The actual location of that gene on the chromosome is called the locus, or loci for plural. And the members of each pair do not have to be identical. So, like I was saying before, you might inherit one hair color from one parent and a different hair color from the other parent. They have the 2 different alleles. Down here I've got a picture of eye color, that's another example. So the alternative forms are called alleles, and for every trait you have 2 alleles for that trait or for that gene, and how they interact with one another will determine what that trait ends up looking like, which we'll talk a lot about in the next chapter, in Chapter 3. So diploid versus haploid, like we were saying during the formation of gametes, you're going to reduce the number of chromosomes by half to produce a haploid cell. So in humans, we have 46 chromosomes in our diploid cells. So our haploid cells are going to have 23 chromosomes. And meiosis is the type of cell division that leads to the formation of these haploid cells. So this is just kind of showing you the process of sexual reproduction. So our sex cells undergo meiosis, or we have diploid cells that undergo meiosis to form our sex cells, which is the sperm and the egg. And each of those are going to have half the complement of chromosomes as our diploid cells, so they're going to have 23. And then during sexual reproduction, when the 2 meet, when you have an egg and a sperm meet, they fuse together to form a diploid cell, which becomes a zygote, and so that zygote is going to be diploid and have 46 chromosomes, and then that zygote develops into a human baby. Alright, so that's it for this first lecture You can answer some of the questions that are on Moodle, and then we will move into the 2 different types of cell division and go into more detail about that, starting with mitosis.